here is the linear case increase, cumulative cases uh, on a linear graph. But if you look at this one here, it shows you the logarithmic scale for the increase in number of cases or cumulative cases. And what you will notice immediately that this curve can be divided into an early phase where there was very rapid growth. And that is when the outbreak was really speeding up in China, especially in Hubei province and in Wuhan. But as China started to control the outbreak, the curve started leveling off. But then it took off again. And the second rise was fueled largely by United States and countries such as Spain and Italy and UK. And now you see that the curve flattened out a bit again, but continues its upwardly rise. And now the outbreak is really growing in our own country as well as some of the other countries in the Asian region. The graph below shows you the daily case numbers, and you will see that again the daily case numbers rose very quickly, and this happened in the first week of March, essentially the first 2 weeks of March, and then they seem to have leveled off a bit, but again the number of cases, at least towards the middle to end of May, seem to be picking up again. And as I said, this is being fueled largely by the outbreak in India and some other Asian countries. What is the status in India? In India, as of today, we have about 1.9 lakh cases, confirmed cases, 5,400 deaths, about 50%, little less than 50% of people have recovered from this infection. And if you look at this graph below, the green line shows recovery, the orange line shows death, and you will see that about 94% people have recovered as of today. About 94% people are recovering. That's the current recovery rate. So with the death rate of India in India around 3%, that is not surprising that greater than 95% people are recovering. And that's one key fact about this virus, that no matter what you hear, the scares that you hear, the recovery rate is pretty good. And this is across the world. And eventually, this recovery rate should approach about 97% because the mortality rate is about 3%. And again, looking at the cases in India on a logarithmic scale as well as on a daily basis, you will see that the cases are increasing. But the slope over here is not as steep as the slope over here, which essentially tells you that since the lockdown happened on 25th of March, the rate of growth of the outbreak has slowed down. So at the time of the lockdown, the outbreak was doubling roughly every 5.5 to 6 days. Today, the outbreak is doubling roughly every 12 to 14 days. So it has slowed down, but by no means has it flattened out. So that's the other key fact that you should understand about India. And this reflects in the daily number of cases, whether you take it on a 3-day average or on a 7-day average, we still have a growing outbreak. We have not reached the peak yet. It is expected that the peak will hit sometime towards the end of June or maybe by the second week of July. And you can imagine where this graph will be at peak. It is estimated that if the outbreak continues to grow at this rate in India, we may see something like 40 lakh cases uh, at peak time. I'm sorry, not 40 lakh, about 4 lakh cases at peak time. But then there would also be a downward slope of the peak, and there would be cases at that time also. So by the time the outbreak really dies down in India, we can have many more cases than we have now. The graphs below show you the death rate, and again, the death rate pretty much mimics the number of cases in India, both cumulative death rate as well as the daily death rate. And you can see that it mimics very closely the cases. So the next question is, where did this virus really come from? Now, to understand that, we have to go back and see where other similar viruses came from. And this I'm giving you a pictorial of all other coronaviruses. So this is the 7th coronavirus, and coronavirus is a family of viruses. It's a family of RNA viruses that infect not just humans, but many animals as well. But this is the 7th coronavirus that has been found to infect humans. Before that, we've had viruses such as 229, NL63, OC43, and HKU1. All these have become endemic to the human population, and these roughly cause anywhere from 20 to 30% of the common cold that you get on an annual basis. The 3 most recent coronaviruses are the SARS coronavirus, which emerged in late 2002, and really by the time it was controlled, it was almost 2004. So it took like a whole year to control. But in the end, it caused only about 8,000 some cases and about 750 deaths. The second coronavirus lately to cause disease in humans is a virus called the MERS coronavirus. MERS stands for Middle East Respiratory Syndrome. And this virus caused many fewer infections, so about 2,500 cases, but you will notice that it caused almost as many deaths as the SARS coronavirus. So what does this tell us? Firstly, it tells us that the SARS coronavirus, which emerged in 2003, had a fatality rate of about 9%, 9.5%. The coronavirus we're looking at today has a global case fatality rate of about 7%. In India, we are seeing about 3%. The MERS coronavirus had a much higher fatality rate compared to the SARS virus. Now, the question is, where did this come from? And the 2 earlier viruses both emerged from bats. And it was shown that the SARS virus jumped from bats into humans through an intermediate host, which was called a civet cat. It's a mammal. And the MERS coronavirus jumped from bats into humans through another intermediary host, which is a camel. Now, there is pretty good evidence now that the coronavirus 2 jumped into humans through another intermediary host, which is called a pangolin. It is also a mammal. And we know this because the closest homology in sequence to the COV2 virus comes from a bat virus and a pangolin virus. The similarity between the SARS-1 and SARS-2 is only about 80%. Between MERS and SARS-2 is only about 55%, whereas between the bat virus and COV2, it's about 96%, and the pangolin virus and the human virus is about 91%. So, and based on few other features, we know that this is the jump that has taken place, and the virus has adapted itself to the human population. But mind you, it is still adapting, and that is why it is causing so much disease. The earlier viruses that have become endemic to the human population only cause very mild disease. Now they have adapted. And one hopes that the COV-2 virus also in due course will adapt to the human population and it will modulate its virulence. But that will take a few years to come. Now, uh, about 35,000 genomes from around the world have so far been sequenced for the COV-2 virus, including about 360 isolates from India. And on this map, I simply show you the Indian isolates, and you will see that the Indian isolates are distributed all over, except that there are two major groups or two major clades in which these viruses cluster. One is this clade over here, and the other is this clade over here. And this kind of analysis sort of tells us how related is the virus circulating in India to viruses circulating elsewhere in the world. And based on that, we can draw a map of possibly where the virus in India came from. And although most people would think that the virus in India came from China, that is not the case. Very few viruses in India, maybe early on over here, these two came from China, but the rest of them, some came from Europe, some came from US, and we have a lot of strains that have come from the Middle East into India. And by the way, this is never a one-way street. If India, the viruses circulating in India have also gone to other places. So it would be wrong to say that we have only been recipients of the virus and we have not given the virus to other places. So that's what the situation looks like today. Now, let us try to understand two things. One is infectivity of a virus, the other is lethality of a virus. And what I have done here is plotted lethality on the y-axis and infectivity on the x-axis. The infectivity essentially tells you how many people can get infected from a single infected individual. And that is also sometimes referred as a number called the R0. The R0 for the SARS-2 coronavirus is somewhere around 2 to 2.5, which is very similar to SARS, little more than the common cold. Common cold is about 1.3 to 1.5, but substantially less than other viruses such as measles, which has an R0 of 15, chickenpox, which has an R0 of about 8. So this virus transmits almost equal to the earlier SARS virus, better than the common cold virus, but less than measles and chickenpox. As far as lethality goes, this virus is killing at the rate of about 5 to 7%. But mind you, that's the case fatality rate, which means that these are this is the percent of people who are confirmed positives who are dying. But since we are not testing everyone, we don't really know how many people overall have the infection. So it's important to distinguish a case fatality rate from something called a population fatality rate. Okay. So the case fatality rate of this virus is about 5 to 7%. In the case of bird flu virus, for example, you will see that the bird flu virus transmits very poorly, but has a lethality of about 50%. So about 50% people who are infected with the bird flu virus don't survive. And This is a virus that that circulated in the world in 2005 and caused a lot of deaths. All right. The other thing is that many people will tell you not to worry because this is just like a common cold virus. And I want to show you data to show you that this is not a common cold. This is not your seasonal flu. If you look at seasonal flu, this is seasonal flu. This is the mortality in seasonal flu. The mortality is in the range of about 0.1 to 0.2%. This is the case mortality we are seeing in COV2. If you look at the mortality on a population basis, you'll still find COV2 to be possibly killing at about 0.5%, which is still about 5 times more than the common flu. So it is not the common flu. The other thing about common flu is that there are medications for that. There is a vaccine for it. For COV2, there is neither a medication right now nor a vaccine. So we have to use what are known as non-pharmacological interventions, which is wearing a mask, physical distancing, all of that. All right. Let me tell you a little bit about the virus. So that's what, this is a graphical representation of the virus. It is something which is called an enveloped virus, meaning that the core of the virus has an envelope surrounding it. And the envelope is made up of fat globules or lipids. And that is why when you wash hands with soap and water, you destroy all the virus on your hands, because as you know, oil gets dispersed in soap, so the integrity of the virus is destroyed. In this membrane, we find 3 proteins. One is called the spike protein, shown in red. This is the protein through which the virus binds to its target cells. And there are 2 other proteins. One is called a membrane protein and the other is called an envelope protein. Now, going further into the virus, one finds that it has an RNA genome. And this genome is very tightly packaged with a protein which is called the nucleocapsid protein. And this really forms the core of the virus particle. And surrounding this core is the lipid envelope, which has the 3 proteins embedded in it, as I told you. The diameter of the virus is roughly 100 nanometers. It is estimated that about 1 ml of sputum from an infected patient contains about 10 million virus particles. And this is where it's important to understand when people say that high temperature is going to neutralize the virus. At best, temperature above 40 degrees is going to neutralize only about 40% of the virus. So you can see how many viral particles will still be around at temperatures that are prevalent or around India these days. That's the viral genome. It's a RNA, produces multiple proteins. And as you know, when the RNA gets into cells, it gets translated into proteins. And that's what happens here as well. The viral genome synthesizes anywhere to from 24 to 27 proteins. There is also, proteolytic cleavage of some of the polyproteins, and I will come to that in a minute. The other important thing to understand is the mutation rate of the virus. The mutation rate of this virus is not very high. The mutation rate of this virus is only about 10 to the minus 3 nucleotide per year, which is very slow for an RNA virus. And we know the reason for this. Most RNA viruses do not have any enzyme that can correct mistakes made during replication. Coronavirus has an enzyme that can do that, so it can repair the mutations that occur in the genome. So just to give you a comparison, this mutation rate is roughly 1,000 or 10,000 times slower than what you see in a flu virus. So coronaviruses don't really change that quickly. So again, when people tell you that this virus is changing very quickly and it will become a harmless virus soon, that's not true. There's no indication that that's going to happen for the next few years. And finally, if you look at the replication timescales, the virus stays uh, in your throat or in your nasal cavity for about 10 minutes before it gets into the cells in these spaces, and it multiplies for about 10 hours within the cells. And each virus particle can produce up to 1,000 new virus particles. So again, when people tell you that if you gargle with hot water, you will protect yourself from the virus, you'll have to gargle uh, every 10 minutes on an average to be able to do anything to the virus. So again, while it's good for hygiene, it's really not a remedy. Okay, if we now look at the virus life cycle, the virus binds to the cells through its spike protein. The spike protein contacts a protein on the surface of cells, which is called ACE2. ACE2 is the receptor, and this interaction leads to the entry of the virus particle into the cell. This can be inhibited by antibodies that are specific to the spike protein. Once the virus is in a subcellular compartment called the endosome, the nucleic acid can only be released if the viral membrane fuses with the endosome membrane. And this is a process that requires the pumping of protons into the endosome. And this is a process that is inhibited by drugs like hydroxychloroquine. And that is why the initial thinking that hydroxychloroquine may work as a drug. Now, once the RNA is released, the RNA produces proteins. The RNA is also replicated into more RNA molecules. And it is this replication which can again be the target of some drugs. The proteins that are produced can be proteolytically digested. And this again is the target for another class of drugs. Now, once enough viral proteins are made, once enough RNA is made, it packages into new virus particles that leave the cell. Here are the four classes of drugs that we're talking about. We're talking about neutralizing antibodies that act here. We're talking about hydroxychloroquine, which potentially acts here. We're talking of replicase inhibitors, drugs such as remdesivir and favipiravir that act here. Remdesivir was developed as an Ebola drug. Favipiravir was developed as an influenza drug. Both of those viruses being RNA viruses, it is thought that these drugs may work here. And there are trials that are going on for all of these. And for protease inhibition, there are drugs such as ritonavir and lopinavir, which were developed originally for HIV infection, also appear to show some efficacy. So this is really the basis for why various drugs are being tried. The virus really, once it replicates enough in the throat, gets down into the respiratory tract and goes into the lungs. And that is where it causes the most amount of trouble. It causes a symptom which is called pneumonia. Pneumonia is simply inflammation of the lung. And what essentially happens is that the virus goes and lodges itself at the very end of the lung in cells that are called alveoli, which are air sacs, which also help oxygen exchange from the blood. And when the virus infects these cells, the immune system tries to control the virus. So there is essentially a fight between the immune system and the virus, which leads to other kinds of cells getting into the lung And this causes inflammation, retention of water, and essentially what happens is that these air sacs get filled up with fluid and prevent good oxygen exchange. And that is why people feel short of breath, they run low on oxygen, and eventually this leads to death. So if we can prevent pneumonia, we can prevent a lot of the mortality and morbidity that is seen with this virus. A little bit about testing for COVID. Testing has come under a lot of discussion, and there are basically two types of tests available. One is something called a PCR test, and the other is called an antibody test. So the PCR test detects the genome, and the PCR really becomes positive a few days before symptoms, and you can see the blue line here or the red line here. And this continues for about 10 to 11 days post symptoms. Now, this is a very interesting phase. This is a phase where people show no symptoms, but virus can be detected in their nasopharyngeal swabs and in their respiratory tract, which essentially means that an asymptomatic person is able to transmit the virus to others. And that is why this virus has really spread so quickly around the world that before somebody gets symptoms, before we can isolate them, that person has already transmitted the virus to other susceptible people. Okay, so it's important to understand that PCR detects the genetic material, and this is the gold standard. This is the most confirmatory test. The other kind of test is an antibody test, and which is shown by these lines here. The purple line shows IgM, the green line shows IgG. But you see that the green and purple lines come up only after about a week to 10 days of symptoms. So if you were to test for antibodies in this period, antibodies would come out negative, even though somebody would be infected. Now, this is the most common type of antibody test which is available. It is called a rapid test. And on the rapid test, there is a cartridge. In this, you put a finger prick blood, add a buffer to it, and simply by capillary action, everything moves down, and you get certain bands which tells you A, whether the test has worked or not, and B, whether somebody is IgM or IgG or both positive, which is shown here. So this is a common line. This is a control line. If this line doesn't appear, it means the test didn't work. So the interpretation is the test worked, but this is a negative sample. This person has no antibodies to the virus. The interpretation here is that the person is IgM positive, but IgG negative, which essentially means that this is an early infection because IgM comes a little earlier than IgG. This means that the person is IgG positive, but IgM negative, which essentially means that it is this person who has a past infection because IgM is negative, IgG is positive. And if somebody is positive for both IgG and IgM, that person is possibly in this phase, which means that it's a recent, possibly an ongoing infection. So that's how you interpret these tests. But mind you, antibody tests are not confirmatory. PCR is the only confirmatory test available to us. But antibody tests help us to screen populations to see how far the virus has gone into a population. There are a number of drug trials happening right now, and I've shown some of them here, just to show that a lot of drugs for other viral infections are being tried out for COV2 because there is no specific drug that has been developed against COV2 so far. Drug development takes time, and therefore people are trying to repurpose drugs for other viruses to see if they work here. Already I can tell you that hydroxychloroquine, which was part of a WHO trial, has been taken off the trial because it was shown that it caused uh, more people who were taking this to become sick compared to controls. In India, this is still going on in high risk healthcare workers. Okay. The other thing is about COVID vaccines. Now, this is where really the technology that is available today has come to good use. Within a few months of the first sequence of the virus becoming available, there are over 130 vaccine candidates that are already in the pipeline. 6 vaccines are already being tested in humans, shown on the table here. And 2 of these vaccines have already completed phase 1 trials with good results and are moving into phase 2. It is expected that a vaccine may be available in the next year or so, but then that doesn't mean that everyone who needs a vaccine will have access to a vaccine in about a year, because as you know, the vaccine has to be manufactured and uh, it's safety tested of every batch and all of that. But it's a very good sign that within a few months, so many vaccine candidates are available. And this is just to show that all kinds of vaccines are being tested. We are testing live attenuated vaccines, meaning that these are live viruses which have been weakened by some means. That's more like the pulse polio virus, which has been used to control polio. Inactivated vaccines, you take the virus, you make large amounts of the virus, you inactivate it using a chemical, and then you give it to people. There are viruses that there are vaccines which are made using other viral backbones, whether they are replicating or non-replicating. And there are genetic vaccines, DNA-based vaccines, RNA-based vaccines, as well as protein-based vaccines. So many of them are being tried out. A couple of companies in India, Serum Institute in Pune, Bharat Biotech in Hyderabad, are at the forefront of getting themselves ready to manufacture these vaccines as they come from trials. Okay, the other thing you may have heard of is herd immunity. So herd immunity is an interesting concept. What it essentially means is that if you have a community, let's say there are 100 people in a community, and in if nobody is protected from a new disease, then the disease spreads very rapidly. But if a few people have resistance, then those people essentially act as barriers to the propagation of infection. And the more such people you have in a community, the better it is to protect even people who are susceptible. So it is estimated that for herd immunity in the case of COV2, you would need about 60% of the population to have immunity. And there are only 2 ways you can get immunity. One is by getting infected, and the other is by getting vaccinated. So since there is no vaccine for this virus, can we rely simply on infection to give herd immunity? So as you see, 80%, about 60% people, the red dots here, would have to get infected to show herd immunity. Now, let's see whether herd immunity is a practical concept today or not. You have all seen how devastated New York City, London, Barcelona, Madrid, Wuhan, they have been. So, for example, New York City had about 16,000 deaths. London had about 7,500 deaths. Madrid about 8,500 deaths. Barcelona about 6.5. Stockholm, which is really, Sweden is advocating herd immunity. They've already had about 2,200 deaths, but only 7.3% people in Stockholm have antibodies. New York City has had about 16,000 deaths, only about 20% people show antibodies. So the point is that if we wait for herd immunity, the death numbers are going to just skyrocket. And therefore, it is good for us not to be isolating ourselves, to be distancing ourselves, so that we can uh, extend the time taken for infections to happen, which will not overwhelm the hospital system in any region, in any country, and therefore people will have access to medical care. That's the whole point about this. So herd immunity today is really not a very viable concept. And just to explain about India, uh, India has a population of 138 crores. If 60% Indians were to get infected for herd immunity, that would be about 83 crore Indians. And if the population mortality is 0.5%, then we are looking at about 40 lakh deaths before herd immunity can take control. And that is precisely why we can't really wait for herd immunity. The idea is to delay as long as we can so that we can have drugs available and we can have vaccines available. Okay? Now, to end, let me just show you some of the infographics that we have prepared. These are available on our website from the Resource Hub. They can be downloaded. They are in multiple Indian languages, and they can be used freely by anyone who wants to really educate themselves and educate others about this virus and this disease. Uh, Let me just finish with some key messages. The first key message is to really follow public health guidelines that are provided by central government, by state governments, and by WHO. Each one of us should behave as though we are infected and we have to protect everyone around us, which means that when you are in public, wear a mask. And that mask can be a simple double or triple layer cloth mask, something which you can bring home, wash with soap and water. That's the best kind of mask that we can wear as common people. We're not healthcare workers. Healthcare workers need a special kind of mask, which is called a N95 mask. We don't need that. Okay. So if you are in public, wear a mask. And mask can be made at home very easily. You can make it out of a handkerchief very easily. The point is you have to wear a mask. There are some other myths. One myth circulating is that there are strong and weak strains of the virus circulating around the world. Especially in India, people say that in India the mortality is lower than the world, and therefore in India we have a weak strain. There is no evidence of weak and strong strains. We have to consider every strain as a pathogenic strong strain. The second is that chloroquine or hydroxychloroquine can be taken as a preventive. Again, there is no evidence of this. In fact, the data that is coming out now is that prophylactic use of hydroxychloroquine may give you other health problems. So please don't go around taking hydroxychloroquine as a preventive. It is not a preventive medicine. The third myth in India and Africa is that past malaria infection or vaccination with BCG, which is a TB vaccine that we get at birth, protects us against COVID. Again, there is no evidence of this. In fact, there are countries like UK that stopped BCG vaccination only about uh, 15 to 20 years back. So there are enough people in UK who have exposure to this vaccine, but they are getting infected and they are dying just like anybody else. So there is no difference. So there is no evidence that these work. And finally, a myth going around is that summer temperatures, especially in the mid 40s to high 40s that we see in India these days, are going to end the pandemic in India. There is no evidence of it. In fact, there are two studies, one from WHO and one from Hong Kong, which are both published in very prestigious medical journals. Very thorough studies have shown that the virus can survive at 37 degrees for about 3 days. It can survive at 56 degrees for about 6 hours, and it can survive at 70 degrees for about 30 minutes. So that so it really, these are not achievable temperatures. So don't fall into this trap. You have to protect yourself. So the bottom line is don't wait for a drug or a vaccine. It is up to us to break the transmission. And the best way to break transmission right now is to wear a mask in public, use hand sanitizers, and even better than that, wash your hands frequently with soap and water. We have just come out of a lockdown. We may go into other lockdowns. Lockdowns are difficult, especially for vulnerable people, people whose livelihoods have been affected. So be kind. It's up to us to stop the virus while protecting people from other kinds of problems such as hunger uh, and starvation. Okay, so I will end here, and the final reminder is that the next talk in this series is going to be by Shubhadar Chaudhary.
Excuse me, sir.
Thank you very
Let's
much.
thank you so much, sir. Uh, people have some questions. The first one is like we have been hearing about various vaccines that countries are trying to make. You also told about a lot of vaccines, around 130 of those. So, what's the scenario of India in this direction? How is India, uh, like where India stands in the vaccination?
Well, there are about 30 vaccines under development in India, but these are at very early stages of development. They are not at a point where they can be tested either in animals or in humans. This will take at least 6 to 9 months before any vaccine from India gets to that stage. On the other hand, there are companies in India that have very good capacity to manufacture vaccines. And already these companies have formed very key partnerships with uh, other countries that are developing vaccines. So you can be assured that if a COVID vaccine has to be made cheaply, and if it has to be made at volumes where the world can get access to it, it will have to be made in India in one of these companies or more than one of these companies. So we are right now looking at manufacture of vaccines in India, maybe at a later stage also. Some vaccine developed in India will also reach that stage.
Okay, thank you so much, sir. The next question is: are people with more allergies susceptible to pneumonia induced by COVID-19?
There is no evidence of it that if you have allergies, you are more susceptible. Uh, but definitely, if you have allergies, and some part of your nasal mucosa or your throat mucosa would be damaged because of the cough. So you would be more susceptible. But there is no evidence that you'll have a disease that is any worse uh, than somebody who doesn't have allergies.
Thank you so much,
Okay,
sir.
sure, sir. Thanks a lot, sir, for those explanations. We have a couple of more questions. So for the next question, someone has asked that what do you think were the main reasons for COVID-19 becoming a pandemic, considering that there were outbreaks such as SARS-CoV or the Ebola virus earlier?
Yeah. Uh, a couple of things. One is that it has a fairly decent transmission rate. If you recall, I told you about a factor called R naught, which is about 2.5 to 3 for this virus, which means that every infected person can infect another 2 to 3 people. So that's one. The second is that unlike the first SARS virus that came in 2003, where people were shedding virus or transmitting only when symptoms appeared, in this virus, shedding is happening a couple of days, maybe about 2 or 3 days before the first symptom appears. So there is no way people would know that they are infected and they go about their normal business while at the same time they're transmitting. So that, I believe, is the key factor why this virus has spread around so quickly around the world. And of course, the world in 2020 is far more connected than the world in either 2003 or 2012. There are far, far more flights. There are more dense supply chains. All that contributes to movement of viruses.
Okay, so thanks a lot for the explanation. We have one last question before we end this. So it's quite an open-ended question, but someone has asked that do you think there'll be more cases in India than what is predicted to be there at the peak as of now? And if yes, how much deviation do you think there could be possibly from the deviated value, from the predicted value? I'm sorry.
Look, I mean, it's not just India. I think everywhere the number that you measure will always be lower than the number you have uh, in real terms, because you never test to saturation. Uh, in India, we are currently testing about 2,600 per million population. So we are, you know, very, very low on the saturation count. So definitely, I think there are many more infections than we are seeing. The, the question is, how much more do we have out there? Uh, it's hard to say, but let us look at the death count. Let me go back in slides. Okay, if we look at the death count, today in India you have 5,400 deaths. Let's say for simplicity, we have 5,000 deaths. Now, if we know that the mortality in the population is 0.5%, then if I multiply this number, 5,000, by 200, that gives me the number of potentially infected people. You got that? So 5,000
Yes, sir.
multiplied by 200. That would be
Around a million.
about 100,000, right?
Yeah, sorry.
5,000
Uh,
multiplied by 200. It's a million. You're right.
it's a
Yeah,
million.
it's a million. It's a million. So based on this death rate, I can back calculate, say that there are 10 lakh cases. Now, that 10 lakh is not today, because the cumulative death figure I'm seeing today is from infection that happened on an average 18 days ago, because that's how long it takes for somebody to go from getting infected to dying or recovering. Okay, so this number was there 18 days ago. So we are possibly looking at about 1 million cases 18 days ago. And if the outbreak, let's say, is doubling at a rate of about 14 days, then today we have somewhere in the range of about 2 to 2.5 million cases. But we are only detecting about 200,000 cases. So that means that we are detecting only 1/10 of what is possibly out there. That's one very crude way of calculating. I won't put a bet on it that there are, 2 million cases today, 2.5 million today, but it's one way of calculating. We never know exactly how many cases are there unless you really start testing for antibodies, but that will have to happen a few years from now to tell you how many people actually got infected during the pandemic. So that's my
Sure,
answer to that.
sure. Sir.
A,
Thank you for that
a rough
response.
estimate is multiply the number you are seeing today by about 15, and you will get an approximate figure for India.
Sure, sir. So thank you for those for all
But
that information.
let me just say one
Yes,
more thing. I
yes, sir.
don't
Yes,
want
sir.
to alarm everyone, because if we have 2 million cases today, remember that the mortality rate is only 0.5%. Okay, so 99.5% people will recover. About 80% people will not show any symptoms. That's a fact about this virus. Okay.
Okay, sure, sir. Thank you for sharing all those knowledgeable insights. I'm sure it helped clear a lot of queries from a lot of people's minds. Perhaps something that people thought that this works and it was a hoax or something like that. And with that, we end the talk for today.